thank you all for coming, and thanks to the Lake Huron Committee for the invitation. The program title was about Lake Huron genetic stock identification studies. I guess I'd like to develop this theme around two different projects. One was a project in collaboration with a number of members from the Quantitative Fisheries Center at Michigan State University to develop simultaneous stock and age-based estimators to estimate stock-specific recruitment from a mixture of animals, mixture of different stock strains, populations, and some applications, and I'll go over a little bit of that. And then an application of a little bit of a different approach, but to focus on the recruitment of wild lake trout in Lake Huron. And I'd like to acknowledge the co-PIs and co-authors, Travis Brendan, Jim Bentz, Weihai Liu, Ihab Tisse, from the Quantitative Fisheries Center, and Jeanette Koneski in my lab, and for this study, similar suites of characters, Travis, Jim, Ihab, and Wendy Lee from the Great Lakes Science Center. So the talk will be in three parts. First is just to provide a bit of background on what is genetic stock identification and the approaches that were developed by personnel in the Quantitative Fisheries Center, and then some information and some how these different models, which are focusing on different ecological characteristics of the different species, have been applied. And I'm going to preempt the Lake Michigan talks by focusing on some studies we've been doing in Lake Michigan, but this certainly has for lake sturgeon applications across the Great Lakes, particularly in the context of some of the juvenile assessment programs that have been on way, et cetera, where age or length at age is available as well as stock. And then I'll talk some of the work that we've been doing with Lake Huron. And so the idea of this simultaneous stock and age estimator goes something like this. So say you have different stocks. These could be hatchery fish, these could be wild stocks, and they are originating, reproducing, and being stocked in different areas of the lake. These animals are of different size and different age. And you have an assessment someplace off. It could be in a standard research, it could be a commercial, it could be a sports fish assessment. And based on a sample of individuals from different strains or inter-strain combinations and the various sizes of these fish, it would be nice to develop a stock-specific recruitment estimate based on these samples that didn't necessitate sampling at a particular spawning area. And oftentimes, historically, these objectives have been addressed using relatively standard methods of marking things and recovering things. What I'd like to offer today is that you can use genetic data to the same ends, and importantly, when you're dealing with moving beyond hatchery stocking to when you have wild animals aren't born with coated wire tags in their noses, nor do they have adipose fins clipped, and you really could use genetics data quite profitably. And so apart from tagging red or yellow fish with different colors, if there's some degree of genetic difference in the populations, the markers should be able to pick this out. So this is an extreme example. We can quantify the variance. This is the variance in red or yellow alleles using some measure of standardized variance. And so even if populations are moderately differentiated, where the frequencies of yellows and reds are different, and thus this measure of variance is considerably different, you still have a means of apportioning out proportional contributions of fish from these different populations, which are contributing to a mixture. So in a hatchery situation where you have fish that are marked in various ways, it's really very simple to identify proportional contributions of yellow. This is an example for the basin, Ontario versus Michigan. On the other hand, if you have wild fish, really the coated wire tags and the adipose fin or other specific strain-specific fin clips really don't provide much inference. However, you can estimate the proportional contributions, and in this hypothetical example right here, and importantly, you can estimate the frequencies at which these different strains are intermixing. And then armed with that data, you can ask some really interesting questions. Well, conditional on the proportions of fish that are stocked of each strain, estimates of strain-specific survivorship, age-specific fecundities, the stocking locations, migration, and these things are the observations that you have right here of size and age consistent with what is expected based on some model. And so the data that I'm going to be presenting today are for 3 different species, and these species really form a continuum about the quality of the age data that are available. So for lake sturgeon, it's very difficult to age a lot of fish, typically based on annuli of the pectoral fin here, whereas, so oftentimes we use length at age, and this is just a figure from Rob Elliott and Brian Gunderman's work of length as a function of age. However, what we're interested in is estimating age as a function of length, so we're kind of tipping this thing around. However, for the other species, we actually have structures, and those structures provide for every fish a much easier way to estimate age. So typically in mixed stock analyses, these are used ubiquitously in marine habitats to manage ocean fisheries, ground fish, salmonids, et cetera. They're used comparatively less frequency in the Great Lakes basin, and oftentimes they really don't incorporate an age. They're just interested in stock-specific contributions. And the analysis goes simply like this. So first of all, sort of a ball and stick view of it. Say you have each of a series of spawning populations and each of a couple of tributaries that are contributing to the mixtures in one of the lakes. You sample each of the 4 populations for, say, 6 loci, and each of the different colors here in the pie diagram represent the different frequencies of the alleles for each of 6 loci for each of these different populations. And you could just see the differences in the frequencies and the chunks of the pie that these populations are different. And so take an example here, and this is a Wisconsin-Michigan, Lake Michigan example. Say you have 2 different loci. You have a color loci where you have sturgeon that could be orange and green, and you have another locus which would be an alphanumeric locus where you fish could be either M or W. Now if these loci are independent and you have an observation for each of the baseline populations of the frequencies of oranges and greens and M's and W's, and these are independent, you can estimate the joint probability of observing different combinations of individuals with genotypes reflected by both loci as just the product of the observed frequencies of each one. And then you can see here that there's quite a bit of difference in this hypothetical example, and you have a mixture which seems to be a mixture of both of these. It really doesn't look like one or the other, and you're interested in estimating the proportional contribution of Michigan and Wisconsin, which is most consistent with that. So that's the ball and stick version. Put it in the form of a likelihood. You're interested in looking at what is the likelihood of the genotypic composition in your mixture, which is a function of or conditional upon the genotypic frequencies in each of your different stock mixtures. And so we have P sub i's, and we're interested in estimating the frequencies of P sub i's, which is a function of the numbers of individuals M, the numbers of stocks, and the genotypes and the stock-specific genotype frequencies. And this is just how the data can be arrayed. So we've done this across all of Lake Michigan for quite a few fish samples. And so you can see a couple of things here. So the different colors in these pies relate to the proportional contributions of these Wisconsin and these, well, it's not Ontario, it's Michigan populations. Okay, so over the entire Lake Michigan, most of the fish are either from Oconto, Peshtigo, or Menominee, but there are some non-zero contributions of these fish over here. You can see that almost all the fish sampled in nearshore waters of Michigan are of Michigan origin. Almost all the fish in Wisconsin waters here of Green Bay are of Wisconsin origin. These little arrows represent, in a couple of the sampling areas, areas where we have representation of the other states' individuals in non-zero frequencies. And so oftentimes you hear about sturgeon migrating great distances, trans-basin migrations, and because the fish can do that, you have the preconceived notion that this is what they do. Well, quite to the contrary, they seem to stay home pretty well. And they do that on a microgeographic scale as well. So this is data that Rob Elliott and his colleague collected from trap nets all through Green Bay, and they're divided based on a southern, a central, and then a northern area of the basin. And you can see here again the same suite of characters right here as you see a progression from southern to central to northern of decreasing frequencies of Oconto, Peshtigo, and fox and increasing frequencies of Menominee fish. Furthermore, if you have juveniles, you break the samples into juveniles and adults, and this is based on about 120 versus less than 120 centimeters, those fish that were captured in trap net locations closer to the river mouths, the juvenile individuals are showing more fine scale site fidelity than our adults. And you could ask the question based on likelihood ratio tests, are there statistical differences in the stock contributions across all groups? And the answer is yes, north versus south, even central B and central A, et cetera. So the populations are really quite structured. So one of the things that we were interested again is using the age, and we were interested, and this is just another way of looking at estimating strain and age composition to estimate recruitment. But here, instead of just the proportions of the different stocks, we're interested in the proportions of each stock for each age class, okay? And because we only have age and we're estimating age from length, there's some degree of uncertainty in here, so we're trying to estimate the proportional contributions of a given age class, but we're incorporating some degree of uncertainty in this length-age relationship in this joint likelihood analysis. So it's a joint likelihood not only of the ages as well as the different stocks. And doing that for the stocks that are contributing to the lake mixture, and this is just for Green Bay, we can see here that we have an annual change, either increasing or decreasing in stock contributions and the standard error of those estimates. And so from this length at age and genetic stock identification, we're able to look at trends in mixture compositions as well as whether those contributions of the different stocks are increasing or decreasing. So that was the sturgeon model. So back to Lake Huron, we were interested in focusing on questions of strain composition in the Saginaw Bay. So it's been known based on capture, recapture, and tagging studies that some proportion of the fish here in the Lake St. Clair, Detroit River corridor and western Lake Erie make migrations into Saginaw Bay. And so to use an empirical example to develop what we call the walleye model, and this is a work that Travis led that's in press. We looked at different stocks, and these stocks here from the Clinton River, various stocks in rivers and reefs in western Lake Erie weren't genetically distinguishable, so we just looked at a Tittabawassee Saginaw River stock versus the stocks here, and we're interested in proportional contributions of fish of different lengths or ages and different strains. And this is what we saw. So here we have the different comparisons, our sample size, and the proportional contributions of Lake Erie, St. Clair, and Tittabawassee River, and the standard error associated with the Tittabawassee River estimates to the Saginaw Bay recreational open water fishery. So the first is over all years with that sample size, all years, all ages, and seasons, 26.6% of the fish in Saginaw Bay caught by Michigan fishermen were from Lake Erie. If you break that down by year, the estimates are pretty consistent across the 2 years. If you break it down by age classes, there are more younger fish being caught in Saginaw Bay that are of Lake Erie origin than there are in older fish. If you break it down by season, there are fewer Saginaw Bay or fewer Lake Erie fish pre-reproductive or pre-spawning time than late spawning time. And if you break that down even further, these 4-year-old fish, you really, 4-year-old and younger in this age category, you have a really much large differences than the older fish. So basically it appears that the older fish are going back to Lake Erie to spawn, whereas you see a relative similarity in the later time periods after the reproductive season. And so this is really interesting biology. So we have different ages doing different things, which seems to coincide with the seasons and who's spawning where, but it also has some implications for what Jim Fielder was talking about, because if there are a lot of walleye in here, a lot of walleye are not from Michigan. They're being produced in Lake Erie. And then applying this model, so again, we're interested in recruitment, so we're interested in the recruitment of a year class in each of these different populations. And here, we're calling this the walleye model. We have a joint likelihood of the genetics and the age, but here what we have is a parameter which is based on the recruitment as well as the stocks or the age-specific survivorship, which is built into the model based on statistical catch-at-age analysis. And this is the analysis, and this is a paper that we have in prep with Travis Brennan as the lead author. So we have a measure of genetic stock identification, or a joint estimate of stock and age, of the relative recruitment across different year classes in the two different baseline populations. So this is Lake Huron, or the Tittabawassee, and this is Lake Erie. And what we've done is we've also included an estimate of statistical catch-at-age recruitment here, and so the white balls refer to the statistical catch-at-age, and these are our estimates based on a joint stock and age ID. And not, maybe this is the gold estimate, or maybe it's not, but you can see for a number of the age classes there's fairly close agreement. However, there are certain age classes, for instance the 2005 cohort, where they disagree quite a bit. And so here are our estimates. We have confidence intervals about those estimates, and so there's quite a bit of variability in year class recruitment. And again, these are all fish that were captured in the recreational Saginaw Bay fishery. The last example that I'd like to talk about, and this is a slide that G presented in the former talk, was trying to use genetic stock identification and the age analysis of fish to understand a little bit more about what strains were recruiting these wild fish. So we have an increase relative to previous time periods. These red portions of the bars represent the proportions of fish of different size classes being of wild origin and hatchery origin. And the question was, what was the year class and strain composition of these wild fish? And again, they don't have coded wire tags, they're wild, they're not clipped, and so we're using the genetic data to estimate strain composition. We're interested in looking at strain and age composition and whether that differs spatially across different statistical management units, if it differs temporally. So we had early samples in the early 2000s and later samples 2010 through 2012. And then we were interested in determining whether these contributions of the different strains could be related to expectations based on the proportions of the strains stocked, to dispersal or movements after stocking, and to survivorship. And then we were also interested in estimating proportions of interstrain hybrids. And so what we did, this represents the two time periods, so 2002, 2004, 2010, 2012. We were only sampling individuals from 4 to 10 years old in each of these different time periods. And with those criteria, these are our sample sizes for these reporting districts, okay? So the criteria for inclusion were wild fish, the range of ages. All these samples were genotyped at 15 loci, and so between Wendy Lee Stott's lab and my lab, this represented over 1,100 strains, and what we have is the first joint US-Canada standardized database for lake trout for those loci, and we genotyped almost 1,700 wild individuals to use for the analysis. And so here are the sample sizes for the US reporting districts and for the Ontario. Unfortunately, while we had representation in both time periods for these three different groups, we only had OM1 represented for Ontario. So I'm going to get into the actual observed estimates in a minute, but we were interested in developing or using the data on stocking numbers, movements, et cetera, to develop an expectation or what series of parameters would both explain the data. And so this is just a hypothetical example. So we were assuming that age at first reproduction is 7, and that plausibly the fish could live for 7 years. And so the representation of different age classes represented in any given time period, early versus late, represented this range. And just as a hypothetical example here, for the spawners that were contributing to reproduction in this age class, so these year classes were represented in the adults representing ages of this, and these were the stocking numbers of each of the different strains of US and Canada that would be consistent, and this is for the US side of the basin. So we started out with that. Then we were using statistical catch and age estimates, and here this is just very simplified. We were not assuming differences across different strains, but we were interested in these transition probabilities of what is the probability of a fish that lives to age 7, and what are the transition probabilities of fish living to reproduce multiple times. We were also using work that was done by Sarah Alderstein, and again used in the statistical catch and age model. So we have a migration matrix, right, of fish that were stocked in some areas, and what is the probability of being stocked in an area and moving someplace else, right? And so we had these are some of the management areas: Drummond Island, 6 Fathom Bank, some of the quartile management areas or quantify management areas here. So we had that data in. So based on the stocking numbers and based on estimates of age-specific survivorship, which were not estimated to be different by strains, and based on the movement data, these are the expectations, and I'm just going to present, we have the estimates for Canada, but I'm just going to be presenting the US data for these reporting areas. And so for each of the 2 periods, early and late, so 2000, 2002, 2010, 2012. This is the expectation of observing a Lewis Lake, or the expected contributions of Lewis Lake, Seneca Lake, et cetera, for US and Canada into these management areas. And I note here that we see here a positive trend towards an expectation of a positive trend for Lewis Lake, an expectation of a positive trend for Seneca Lake, and an expectation of a negative trend for Marquette, right? That's as an expectation based on stocking, et cetera, okay? And this is what we observe. So this is over all time periods, and this is the standard errors. I didn't report the standard errors, but they're fairly tight confidence around those estimates. But here for the Michigan reporting areas, just synthetically early versus late, and these different management units. And here's something that speaks to an estimate of degree of assortative mating. So this assortative mating coefficient is 0-1 distributed. An assortative mating coefficient of 1 means all the individuals mate with individuals from their own strain. An assortative mating coefficient of 0 means total panmixia, right? You're breeding with 100% probability with somebody outside of your own strain. So it's a 0-1 value. So what do we have here? So in the expectations, we had a plus, plus, minus, and here we actually are observing a decrease in Lewis Lake, an increase in Seneca, and a decrease in Marquette strain. These are the genetic estimates of strain composition. So we see, with the exception of Canadian Seneca strain, and they're really very genetically similar to this, so I'm suspecting that it isn't what Canadian put in there. It's something that looks genetically a lot like what Seneca, what the US is putting in there. But we see relatively little contribution of any of the Canadian strains to US waters. The other thing is that there's a considerable heterogeneity here across the management units and the proportional contributions here. So we see a declining trend in Lewis Lake, but that's really reflected in MH1. We see a increasing trend of Seneca Lake, and that's most evident in MH1. And we see a decreasing, and there's a consistency, although being greater declines in MH1 and MH2. The other really interesting thing is we're seeing between these 2 different time periods more evidence of random mating, less assortative mating. So it's more likely, as we're going through time, for these individuals to be reproducing with somebody other than a member from its own class. And how is that? So we have, in a hypothetical situation again, we have different strains, and they're producing something in the wild. So you have some individuals which are still homozygous for yellow, which means they're breeding within a strain, so likewise these guys, and then you have some proportion which are interstrain hybrids. If you take a look and just with the expected frequencies of the different strains and estimate what is the probability of an individual reproducing with somebody else of its own strain, it's just the expectation of that strain squared, right? So the probability, if strain I is present in probability P, the probability of two individuals mating from that strain is just the probability squared, right? And you get that as an expected probability of assortative mating. And that's largely because of the predominance of some strains with high levels of survivorship like Seneca strain. But what we observe here is really something less, even in the early time period, and we see a dramatic difference as time goes by, and that's even with continued stocking of strains over time. It's really kind of a puzzling thing, and there's several ways to explain that. One way is that with sea lamprey control increasing with effectiveness, you have other strains besides Seneca strain that are surviving, potentially, and there are multiple more strains that are contributing more, and that could be an explanation. Another intriguing explanation is that these individuals in the late time period are being produced by wild fish from the early time period, and thus they're not F1s, which is what we were assuming. They're actually F2s, or wild fish being produced by wild fish. And the slide that G showed of the increase in recruitment of wild versus hatchery fish in the system, or the survivorship that graph he showed, is really very consistent with the latter hypothesis, which I think is really something that needs further attention and is really quite interesting. And I'll leave it for questions.